Dr. Hansen here. A couple weeks ago I gave an introduction to cell signaling, and now I want to look at one cell signaling pathway in detail. What's so nice about G protein coupled receptors is that they're very widespread and medically relevant, and they make a good archetypical system for demonstrating the principles of cell signaling. In terms of learning objectives, I hope you'll focus on interpreting nomenclatures, because you will need to know the names and functions of every component of a G protein coupled receptor signaling pathway. And in fact, I'd like you to be able to sit down and diagram these pathways. I include the Lippincott textbook because they have a brief description of a G protein coupled receptor signaling pathway in Chapter 8 that corresponds to part of this talk. You might want to look at that after you've done the preview video and exercise if you still have questions. It does give a nice overview. As a physician, is it important to understand endocrine signaling? Well, I should point out that an awful lot of deadly diseases result from failures in cell signaling. For example, cancer. That's caused by a mutation that generally will lock one of the intermediates in a cell signaling pathway into the on position so that cells keep growing and dividing. Diabetes. This is a failure in signaling with insulin, glucagon, other counterregulatory hormones. Heart disease. Well, that's correlated with diabetes and metabolic syndrome, which again can result from a failure of regulating energy homeostasis. Finally, a lot of pharmaceuticals target cell signaling pathways. In fact, half of all pharmaceuticals specifically target G protein coupled receptors. Therefore, as you work through this PowerPoint and do the activity in Cougar, I hope you're thinking, not only is this important for the upcoming test, but this is going to be important in neuromuscular when we look at synaptic signaling. It'll be important next semester when we look at insulin and glucagon diabetes. It'll be important next year when we look at the hypothalamic pituitary pathways, which regulate the thyroid, the adrenal gland, the testes, ovaries. In fact, these pathways are not a learning objective today, but you will learn each of them and every step in them next year in endocrine. And since we're looking at G protein coupled receptors today, I'd like to point out that the hypothalamic pituitary axes all begin with G protein coupled receptors. In most of them, the very second step is also signaled through another G protein coupled receptor. Our current understanding about how G protein coupled receptors send a signal from outside the cell to inside the cell was greatly advanced through functional studies by Dr. Lefkowitz and by structural studies by Dr. Kobilka. And I do have to say their Nobel Prize speeches are very entertaining and very informative, but you probably don't have time to click on these links right now. Instead, let's take a look at the take-home message. The take-home message is that G protein coupled receptor structure determines its function. Sounds like AT still to me. In fact, these are transmembrane proteins with an extracellular ligand binding domain, intracellular activation domain, and they're connected by 7 transmembrane helices. In fact, these are frequently referred to as 7 transmembrane receptors or 7 transmembrane helical receptors. And it turns out these transmembrane helices are fundamental to their function. When a ligand binds, it alters the interaction among the helices such as they startly shift around relative to each other, and that shifting of these helices opens up the activation domain on the inside of the cell so that the message gets through the membrane through a conformational change. The ligand itself, the hormone, does not ever enter the cell. And we'll talk about that in more detail later. But first, I'd like to take a look at this huge dendrogram of the whole family of G protein coupled receptors that have been identified so far. You'll notice not only is this a big family of evolutionarily related proteins, but in addition, quite a few of them now have structures. Since the first structure was solved of a G protein coupled receptor, many additional ones have been solved, some with ligands, some without, some with inhibitors. And from all these different structures, many more insights into the working of these machines have been determined. Perhaps with such a big family of receptors, it's not surprising that half of all pharmaceuticals target specifically G protein coupled receptors. The dendrogram itself is branched largely based upon the ligands which activate the receptor. For example, up top here we have my favorite amino acid, glutamate. And the glutamate branch, of course, is going to be very important about a month from now in neuromuscular because it's the most important neurotransmitter in the CNS. Of course, the receptor itself is simply one component of the entire pathway. For example, we also have to include release of the hormone, transport of the hormone through blood, binding the hormone to the receptor, and then all the downstream effects which are caused by activation of the receptor. In fact, every component of G protein coupled receptor signaling pathways that are included in this figure is part of a learning objective. And so you should know the name of every part and what the role is and how the pathway flows. And I'll summarize the overall pathway and then I'll go through each component individually. So here we're showing that the hormone binds to the receptor. And whenever a protein binds to a substrate or a receptor binds to a ligand, that receptor or protein will change its conformation and wrap around the ligand. In the case of G protein coupled receptor, that induces a conformational change in the transmembrane helices, which opens up an activation site inside the cell. The nomenclature is relatively simple, but it can be a little tricky. So what I want you to do now is to interpret the nomenclature to tell me exactly what the G protein coupled receptor is and exactly what the G protein is based on the figure. Please pause and do so, but I will move on. The G protein coupled receptor is shown in purple. It's called a G protein coupled receptor because it's a receptor and it is often coupled to a G protein, meaning the G protein is shown in red. The G protein is called a G protein because it is commonly bound to a guanylate, GDP or GTP. This particular G protein is trimeric, meaning there's an alpha, beta, and gamma subunit, and the GDP or GTP are bound specifically to the alpha subunit. Upon activation of the receptor, the trimeric G protein becomes coupled. The interaction causes a conformational change in the trimeric G protein. That causes the alpha subunit to lose affinity for GDP, which comes off, which opens up a site for GTP to bind, which again induces a conformational change, causing the alpha subunit to separate from the beta and gamma subunits, and the activated GTP-bound alpha subunit now will have high affinity for adenylyl cyclase. What do you think happens to adenylyl cyclase when the activated alpha subunit binds? It'll undergo a conformational change, which will open its active site to bind ATP and to catalyze hydrolysis of ATP to release pyrophosphate, forming cyclic AMP. It's a learning objective to know that cyclic AMP is the secondary messenger of G sub I or of G sub S type of G protein coupled receptor signaling. It's also a learning objective to identify the primary messenger of a G protein coupled receptor. Please pause and do so. Do you agree that one's pretty simple? How about the hormone itself? Primary, secondary. So at this point, I want to go back to the first step in the figure where the hormone binds to the receptor, and as we said, the receptor will wrap around the hormone and undergo a conformational change, shown in more detail here. Here I'm showing two views of G protein coupled receptor. This one I'm looking at a cross section through a membrane so we can see the ligand binding domain on the outside, the activation domain on the inside, and we can see each of the helices. In addition, I'm showing the inactive conformation in orange superimposed on the active conformation shown in green. Also, if we rotate this figure 90 degrees around the x-axis so that we're looking up from the inside of the cell, up the membrane into the receptor, we can now see these same helices end on. What is the biggest change you can see in both figures? Well, most of the orange and green helices perfectly overlap, the greatest exception being the one helix down here on the lower right, or if we look at the end views, we see that this is called helix 6. Please don't memorize that this is called helix 6, it's simply to put a label on it, but do you see how much helix 6 has changed its orientation relative to the remaining helices? What has that done? It has opened up the bottom of the receptor. In the inactive form, there's a small hole at the bottom. When helix 6 moves out of the way, there's a large open cleft in the bottom. This is the active form because this cleft is where the trimeric G protein is going to bind. Another point of interest here is we're showing where the ligand is on the outside of the membrane. It will not get through the membrane into the inside. Rather, it is the conformational change that brings the signal into the cell. In describing how G protein coupled receptors convey various signals from the outside of a cell to the inside of a cell, the teeter-totter model can be convenient. In this model, we're representing G protein coupled receptors as teeter-totters, which can rock back and forth. They have a ligand binding site on the outside, a G protein binding site on the inside. And here I'm showing two different conformations. Based on my descriptions of the structure of G protein coupled receptors so far, can you tell me which one of these is in an active conformation? A clue would be to look at the cleft on the inside of the cell. Can you answer now? I mentioned that the G protein binding site on the bottom was only accessible when the cleft on the bottom opened up, and in the inactive form, the cleft was too small to bind to the G protein. Accordingly, the form with the narrow top and wide bottom would bind the G protein, whereas the form with a wide top and narrow bottom would be the inactive form. And for that matter, in the teeter-totter model, the idea is that the binding of the hormone closes the top and rocks the bottom open. And we'll be using this model in class to talk about some pharmaceuticals. But now I would like to move on to the next step in G protein coupled receptor signaling pathways and look at the trimeric G proteins. To describe how the trimeric G proteins cycle, I want to begin with the inactive form. So floating around, bound to membranes, is the trimeric G protein with the alpha subunit bound to GDP. When a ligand binds to the receptor, the trimeric G protein can bind to it, that then you will lose the GDP and pick up GTP. And, of course, then this can activate adenylyl cyclase. But just like any switch, when you walk in the room and turn on the light switch, you want to remember when you leave the room to turn it off, because if the light is left on full time, your electric bill will go up. Likewise, these activated alpha subunits bound to GTP need to be turned off. Hazard a guess as to what might turn off the activated alpha subunit. We are bound with GTP. We know that the GDP is the inactive form. Big clue. The alpha subunit actually has a GTPase activity. And you're thinking, well, that's kind of wasteful. Not really. You want this to be a short signal. You send the signal. Once it's gone, you can reboot. The GTP is automatically hydrolyzed by the alpha subunit. Now, bound to GDP, it will have high affinity for the beta and gamma subunits and reform the trimer. which is again now ready in case another hormone binds. Every step of these signaling pathways automatically turn themselves off under normal, healthy conditions. And now I would like to go and look at the last step, the adenylyl cyclase enzyme. As I mentioned earlier, binding of activated alpha subunit to adenylyl cyclase causes a conformational change and thereby activates adenylyl cyclase. Most of you are very familiar with seeing this cyclic AMP, CAMP if you will, but I think it's useful to actually look at the structure of this compound. Adenylyl cyclase binds to ATP and catalyzes a nucleophilic attack to form cyclic AMP while releasing pyrophosphate. Please don't get bogged down in this, but I do think it's in your interest to see if you can draw the two arrows to show the nucleophilic attack. What you do is you look at the substrate in the product. Where does the nucleophilic attack have to be? Also realizing that pyrophosphate comes off. Also, where will the oxyanion be? Go ahead and do that. Don't spend too much time. I'm going to move on. Well, I'm looking here saying, okay, this phosphorus atom must be this one. These two must be here. Does that help? And we see that the 2' hydroxyl is over here, which means that the 3' one must be the one that attacks. The electrons resonate up, they resonate back down, and kick off the electrons in this bond onto the pyrophosphate, which can pick up a proton. And I really think that cyclic AMP is kind of a pretty compound. Really nice to have this chair-shaped 6-membered ring. And of course, now I have to ask, how are we going to turn this signal off? All signals get turned off. How do you turn off the secondary messenger here? Please take a stab at that. Well, I think it's pretty straightforward to take a structure like this and nucleophilically attack it with a water molecule. And in fact, an enzyme is going to catalyze that reaction called cyclic AMP phosphodiesterase. Please interpret the name of that. And the final product is AMP. So we've hydrolyzed a triphosphate and got rid of the pyrophosphate, went way down in energy. We've hydrolyzed another phosphodiester bond to make simply AMP. Everything's gone down in energy. Therefore, this AMP could be acted upon by the enzymes in the body which regenerate energy and rephosphorylated back to ATP. And if you want, it could go through the cycle again. At this point, I want to mention that cyclic AMP is a secondary messenger for 2 different types of G protein coupled receptor signaling pathways. They're referred to as G sub I and G sub S. Both G sub I and G sub S types of the alpha subunit bind directly to adenylyl cyclase and alter its activity. Can you tell me which of them stimulates and which of them inhibits adenylyl cyclase activity? It is important that you stop right now and answer that question. The reason it's important is because I want to be sure you see how simple it is and don't have any trouble with it. Of course, G sub I is G sub inhibitory. It'll inhibit adenylyl cyclase, which will decrease the production of cyclic AMP. G sub S is G sub stimulatory. It will stimulate adenylyl cyclase to produce cyclic AMP and raise its concentration. I'm hoping this has sort of helped you understand the pathway and made it easier to remember the nomenclature. But now I would like to look at an important example of a G protein coupled receptor signaling pathway. The man in this image is almost certainly producing a hormone and releasing it into his blood. That hormone acts through a G protein coupled receptor. What do you think that hormone is? Please pause and answer. Well, I'm hoping here you got, what is it? How about adrenaline, epinephrine? And there are receptors on the liver for epinephrine. Those receptors regulate metabolism through many enzymes. Among the enzymes are glycogen phosphorylase and glycogen synthase. Please interpret each of these names and then, based upon the scenario, explain whether or not each of these enzymes will be stimulated or inhibited by epinephrine. Please pause and do so, but I will move on. Adrenaline is a really good signaling pathway to use if you want to understand diabetes, because adrenaline, insulin, glucagon, these hormones all regulate metabolism and are tied in with diabetes. And when you're asked this question, what are the conditions under which adrenaline is released? Under stress. Stress says, I need sugar right now. So knowing you need sugar, you should be able to interpret the names glycogen phosphorylase and glycogen synthase to decide whether or not epinephrine will stimulate or inhibit them. And in particular, glycogen is a source of glucose. Phosphorylase literally means that you link a phosphate to glycogen in order to break off glucose monomers. And of course, glycogen synthase is the synthase that makes glycogen. That is, it links glucose into a polymer. If a bear is chasing you, you don't want to be taking your glucose monomers and linking them into a polymer. You want to be sending them to your muscles to be able to run. And therefore, you want to break down the glycogen reserves. This is going to underlie a lot of renal and endocrine, especially when we talk about metabolism and diabetes. Of course, when it comes to regulating your metabolism, I have a word of advice coming from the rangers at Yellowstone. My family and I were camping there last month. My daughter loves the junior ranger programs, and she learned, don't get within 100 yards of a bear. Good advice if you want to save your glycogen stores. In conclusion, I hope you understand every component of G-protein coupled receptor signaling, at least up to the production of cyclic AMP by a G sub S or G sub I type of G-protein coupled receptor. And then you will be able to understand the activity we're going to have in Cougar this week. This also will be important to understanding what we'll cover next semester and next year. And of course, ultimately, you will be treating a lot of patients with diabetes. This is at the heart of how to think about it. If you have any questions, I hope you stop by even before the activity or ask during the activity or stop by later. Thank you.